assay, such as a co-IP, allows us to look and see what molecules are interacting with what other molecules. We take one of these molecules and we capture it, and then we look and we see what it pulls down with it. And so often we're doing this capturing using antibodies, and so in this case we call it a co-IP. We can also use things like affinity tags, and I'll get into this. So a co-IP is just a form of a pull down, and often with these you're using some sort of magnetic beads. What's going to happen is you're going to be able to isolate the stuff that's bound, wash off that stuff that's not bound, and then look and see, okay, well, what was bound? So here's a lot more detail.
So just an overview, we're going to go over what these experiments are, some practical things about how you actually go about designing and setting up and running these experiments, as well as how you interpret the results. But let's start with the terminology. So you often see the term co-IP, which stands for co-immunoprecipitation. And this is just a form of a pull down where you're using antibodies in order to do that capture step. So with any sort of pull down, the basic idea is that you're capturing, you're like pulling down a protein of interest, or it doesn't have to be a protein. It can be an RNA, it can be a DNA. It just has to be something that you can selectively capture or like bind to little beads. And then what you're going to do is you're going to pull it down along with whatever it's attached to. And so this is where we get the co-IP in the IP. If you were just doing an IP, just an immunoprecipitation, you'd be focused on that initial protein that you're trying to capture, the one that the antibody is actually primarily bound to. But in the case of a co-IP, what you really care about is the prey. You care about the other proteins, the other molecules that are coming along with that initial thing that you captured. And so a co-IP is going to be a pull down where you're using antibodies to do that capture. And as we'll get into, there are other types of pull down where you can use things like a GST tag and glutathione beads. Or biotin and streptavidin. And this is going to allow you to do a very, very similar type of thing. Everything's almost identical, except that that original capture is not using antibodies. It's using some other molecule. In any of these cases, what's common these days is the use of these magnetic beads, which are really, really cool. Basically, you take your sample and you mix it with these beads after you wash those beads a bunch. But the beads, what they do is they're going to provide a way in order to separate the beads from the liquid. So you're going to be doing a series of washes and things like this, where you're going to want to isolate the beads from the rest of the liquid, and pipette out the rest of the liquid. And these beads make it really easy to do it. And so basically how it works is you have these magnetic beads and you mix them with your thing. So you can see that when you stick them on the column, the beads are going to get pulled to the wall. And this is going to make it possible to suck out that liquid. Alternatively, what you can do is use non-magnetic beads, often like agarose beads, and what you can do is you can just do a gentle centrifugation step in order to pellet out those beads and remove the liquid. And so basically these methods are going to allow you to separate the beads from the liquid and therefore separate the bound things from the unbound things. The bound things should only ever be your bait, as well as what the bait's bound to. Everything else should wash off. And then finally, what you're going to do is you're actually going to unbind your bait. So you typically denature it. So this can be with like SDS page buffer, or it might be with an acid or a base or something like this to get your stuff to unbind. And so that's the only time where the liquid you're pulling off is actually going to be what actually has your bait, was going to be in that last step. Initially, it's going to be things that are not bound. And you're going to do a few wash steps because each of those times that you're pulling off the liquid, you can't get all of it. Basically, there's going to be some stuff that's non-specifically bound, and so you want to kind of wash off that really weak stuff. And so these beads are going to make it really helpful in order to do this, these multiple washes. And we'll get more into some of those practical details. But first, I want to get back to some of the terminology and the difference between like a co-IP and the pull down. And so a co-IP, remember, is one form of a pull down, but not all pull downs are going to be co-IPs. And so the IP, the immunoprecipitation, we're basically, the immuno tells us that we're dealing with those antibodies. And when we're dealing with these antibodies, we have a couple of choices. We could use antibodies that are going to target the actual protein, or we can use antibodies that can target a like a tag we put on the protein. And I'm going to be talking mostly in terms of proteins because that's how you typically see it. But you can also do things where you do pull downs based on like biotinylated DNA or RNA, and you pull those down, and then you look and see what proteins are bound or what DNA or RNA is bound. You can do things like pull down proteins and look to see what DNA or RNA is bound to them. There are various variations, but your typical experiment, we're talking about like a co-IP, you're pulling down one protein and seeing what other proteins are bound to it. As we'll get into, there's going to be a couple of different ways that we can actually identify what's bound. If we just want to test for a couple specific proteins, what we can do is a Western blot. And if we want an unbiased approach to look and see whatever's bound without knowing in advance what's likely to be bound, then we can do things like mass spec. So you might see like IPMS and IP mass spectrometry. So sometimes people use the term IP when they really mean co-IP, and the terms can get kind of confusing. But in any case, where you're seeing an immunoprecipitation or a co-immunoprecipitation, we're going to be dealing with capture using an antibody. So let's get back and talk about what those antibodies are for. So you have some options. So an antibody basically is just a little protein that's going to bind to a specific part of something else. And so that specific part of something else is going to be the epitope, and that something else is going to be the antigen. So in my little play example, when we have antibodies recognizing Waldo, Waldo would be our antigen, and the exact site on Waldo where that antibody binds is going to be the epitope. And so the epitope that we're going to be targeting with our antibody can be different. This epitope can be part of the native proteins or part of like the actual protein itself, the unmodified protein. Or what's more common these days is that the epitope is actually going to be an epitope tag that we add onto our protein. So this can be something like an HA tag or flag tag, MIC, V5. Basically, these are short sequences that we have really good antibodies against. Just like that you can commercially buy. And so the epitopes are typically these really short sequences. And so we just have to stick this really short sequence onto the end of our protein. And now we have a site that there's really good specific antibodies against. And this is really important because not all proteins have good antibodies available to buy for them. And even if they do, they're typically really expensive. And more on this in another post, but basically everybody's looking for different proteins. These antibodies, they can be hard to make. They involve often the use of animals, very expensive types of things. And especially for if only a few people are going to actually care about wanting those antibodies against that protein, it's not going to be cost effective for the company to make them. And so often what labs end up doing if they need an antibody against the actual protein is that they'll have to go like pay through a service and actually get antibodies made for that specific thing rather than get them like buy them directly already produced. You have to actually go through this process of getting them made and all this stuff, and it can be really expensive. But if you have some sort of little epitope tag, if you have a flag tag or an HA tag, well, lots of people use those. And so these companies sell you those antibodies, and you can get those antibodies like beads directly attached to those antibodies rather than having to actually attach them to the beads yourself. And so basically, in order to do this, however, you're going to have to do some genetic manipulation in order to actually stick that tag on. In order to stick the tag on, what you're going to have to do is you're going to have to actually add the sequence to make the tag onto the gene for your protein. And so basically, in order to do this, you're going to have to genetically engineer the protein, genetically modify it. This can be done in a couple of ways. One is if you want to actually alter the actual native gene in its native context, so the endogenous gene. In this case, you'd have to do some sort of gene editing with something like CRISPR, and this can be a long and hard process. What people often do instead is they'll actually introduce another copy of the protein, say on a plasmid. They can stick this protein on this in the instructions for making this protein into this plasmid, so the circular piece of DNA, and then they can stick that into cells and get cells to make it for them. This can be done in a variety of different methods, and so often if you're dealing with mammalian cells, say you're using some sort of viral transduction system or whatever, so you're putting in a harmless virus that can then get the cells to make that modified protein. And when the cells are making this modified protein, now this modified protein is going to have that tag and allow you to pull it down. Sometimes people actually knock out or at least knock down, so reduce the expression of the endogenous version, the non-tagged version, so it doesn't compete with things. Other times people will just have this tagged version expressed alongside the native version. And so those are just some technical facts to keep in mind. In terms of the other benefit of getting using an epitope tag is that you can buy these antibodies already bound to the beads. If you're using an antibody that is not bound to the beads already, what you're going to have to do is you're actually going to have to attach it to the beads yourself. And to do that attachment, you typically use these beads that bind to antibodies. So often they're like protein AG beads. Well, protein A and protein G are these bacterial proteins that bind to the kind of like generic part of the antibody. And so you can get these beads that are then going to bind to your antibody of choice and allow you to then isolate your protein. In that case, the binding is going to be non-covalent, so it's not permanent. And so you do have to worry about your antibody potentially coming off the column if your elution conditions are too harsh. And then this can interfere with some later processing, as I'll get into. When you buy these ones like the anti-HA and things like this, they're often covalently attached, so they're like permanently attached to these beads, which makes it less of a concern about those antibodies actually coming off the column. So all of that was dealing with the IP and the co-IP, so where we're dealing with immunoprecipitation, so using an antibody. But the exact same principles pretty much hold true in terms of the protocol for the pull down, where you bind, you wash, and you elute, but binding based on a different property. So this is often also involving the genetic manipulation. So instead of adding one of those short little epitope tags, what we can do is we can actually add a bigger thing, like a protein, like a fusion protein, like GST, we often use GST for improving expression and solubility and things like this, but it can also act as one of these, like an affinity tag, to use affinity capture in order to capture your bait. And so in this case, GST is this little protein that binds to glutathione. So if you have glutathione coated beads, this is going to allow you to capture your protein of interest. Similarly, you can do things like if you want to capture biotinylated DNA or RNA, you can use streptavidin coated beads. There are a variety of different options. And again, all of these have the same principle in mind. We're going to bind that bait. You're going to wash everything else off. Then you're going to elute or kind of like unbind your bait. And then you're going to analyze. So I want to get more into this idea of the analysis. The most common type of analysis you're probably going to see is a Western blot, and we're going to get much more into the details about this. With the Western blot, basically what you're doing is you're taking those proteins and you're going to separate them by SDS page, which is a technique where you denature, so you unfold the proteins, you send them traveling through a gel using electricity. The proteins are then going to separate based on their size, because the longer proteins are going to get tangled up more in the gel mesh, and so they're going to travel slower. And when you turn off that electricity, they're going to be stuck in place in the gel, and the bigger proteins won't have moved as far. You can then take all of those proteins and transfer or blot them onto a membrane. Then basically you use antibodies to probe the membrane in order to determine whether specific things were present. So here again, we're going to be using antibodies. So even if you use a non-antibody pull down method, you're still, if you're doing a Western, you're going to be using antibodies here. These are going to be different than the antibodies that you use in the beginning. And so in the beginning, you're using antibodies that are recognizing the bait protein. And here you're using antibodies that are looking for various prey. The way that a Western blot works, you have to actually, you're only probing for specific things, whatever the antibody that you're using is probing for. And remember, these are the prey antibodies and not the bait antibodies, although you also do a bait antibody just to test and see your efficiency, and we'll get more into this sort of thing. But here, basically, you need to know what you want to look for. And so you're not going to be able to detect any binding partners, any prey that you don't know going in could be prey, so that you don't therefore test for. If you want to see whatever's bound there, you can use mass spectrometry. So sometimes you might see this abbreviated MS, and you might see like IPMS for immunoprecipitation mass spectrometry. And here it gives you an unbiased look at whatever there is. Basically, mass spec, what it does is it takes all those different proteins and things, and it chops them up into smaller pieces. And then it sends these pieces through this machine where it's going to be able to recognize the charge and the mass of the pieces. And by comparing it to a database of the known pieces of proteins, it can then tell you what proteins were present. And so the IP mass spec is going to give you a more unbiased look at what was bound. But the Western is more commonly used, and it's a lot easier, and this is what people are often doing. And so I want to go into detail about how you actually go about interpreting one of these Western blot results. So in a Western blot, we have all those proteins. And if we were to do just like a total protein stain, we would see a lot of bands. We would see something like this. In a Western blot, we're just looking for those specific proteins. If those proteins are present, we'll see a band. If there's a lot of the protein, we'll see a stronger band. Not as much, we'll see a weaker band. But we're only seeing a band corresponding to the protein that we're actually probing for. Typically what you're going to see is you're going to see a lot of different like panels in the gel, in the image, or they're often just like these kind of rectangles on top of one another, where they're showing you that they've probed for different prey, as well as a probe for the bait to tell you about the recovery efficacy, as we'll get into. But basically, you want to be able to test for a lot of different things. I have another post about how you can test for multiple things on the same membrane, so from that same initial gel. But bottom line is you can do this, and so you can then probe for different prey you suspect are present, as well as like do things like probe for something you shouldn't be present to see if you get non-specific stuff, as well as probe for your bait to tell you about the recovery efficacy, so how well it bound to the beads, and we'll get into this. The two main lanes that you're going to care about are going to be your input. So this is going to be what you put in to bind to the bait, and kind of like your pull down fraction. So sometimes you might see this labeled the IP, or the pull down, or the eluate. So basically, elute is where you do that unbinding, and you have your eluate, which is like the stuff that got eluted. And so this is going to be the portion that has the stuff that was bound to the beads. It's really, really important that you remember to take this input sample before you actually bind to the beads. This input is going to be what you're going to compare to your pulled down sample in order to tell you about what portion of it was actually pulled down. So if you, in your pull down lane, you don't see any bands, is that because none was pulled down, because they don't bind, or because there was none present in the initial mixture? Similarly, if you see a strong band in your pull down, is because it bound really strongly, there was a lot of it bound, in comparison to the total, or was it just that you started with a much higher input? And so it's really, really important that you remember to take that input sample before you start your experiments. I should also mention that when you see the input, you often see people do like a diluted version of the input, because there's going to be the signal would be way too high. It will kind of drown out some of the other signals. So people often do load a diluted version of the input. Speaking of the strength of that input bands, basically each of these bands, the strength of it in your input sample is going to tell you about how highly expressed the protein was, or how abundant it was in your original sample. And so with the stronger the band, the more of it there was going in. And this is then going to impact how you interpret the results of your pull down, because you're going to be comparing that pull down to your input. And so what's this going to tell you? In the case of where you're probing for your bait, this is just going to tell you about the recovery efficacy. So basically how well it bound the beads. And note that if you're just like totally overwhelming your beads, you still might have a much weaker band in your pull down than in your input. But basically, if you see a really weak band, this is going to indicate that your protein isn't binding that well to your beads. If you're using your own antibodies rather than using and attaching them to the column yourself, rather than using whatever commercially bound already, you can often get better luck with a polyclonal antibody. So when we talk about antibodies, we can talk about being monoclonal, which only recognizes a single epitope, or you can talk polyclonal, which is going to be a mix of antibodies that are going to recognize different epitopes. So with a polyclonal antibody, sometimes we worry about things like cross-reactivity, but the polyclonal antibody is going to have higher binding affinity. It's going to be able to bind to your protein of interest, to your bait, to your antigen in multiple places. And this is going to allow more for a higher affinity binding. And so you're going to get more bound potentially. And also what can happen is that if you have a monoclonal antibody, so if it's only binding to a specific site, that specific site might be also where that protein is interacting with another protein. And so if you're using a monoclonal antibody against that site, you could be blocking binding. And so sometimes what people do too is they actually might use multiple different monoclonal antibodies, either a mixture of them together or do them separately in order to detect interactions that might have been hidden by one of them. So with the initial binding, with your capture step, you're often using a polyclonal antibody. And then later in the detection step, you're typically using, you might be using a monoclonal antibody, because you want to be really sure that you're detecting what you think you are. And so we'll get more into that. But so that was the bait. But what you care most about typically is this is kind of like one of your controls. So I'm giving you more information about what's going on. But what you really care about is whether or not the prey is binding and how strong the interaction was. And so by comparing the amount of prey in the pull down versus the input, as well as taking into account the amount that you would expect based on the amount of the bait in the pull down versus the input, this is going to tell you about how strong the interaction with the bait protein was. And so if you have a stronger band, this is going to tell you that the interaction was stronger than if you have a weak band, which is going to tell you that the interaction was weaker. And so you can see that in this example, if you had the 4 copies of the gray protein and only one was bound, your pull down band would be fairly faint. Whereas if for this orange protein, you had 3 copies of them and you have 4 copies of them and 3 were bound, you're going to have a stronger band. And so the strength of this band is compared to the strength of the input band, is going to tell you about how strongly they bound. And note too that it's really important that you're comparing it to the input and not to just like another band in your pull down, both because the amount expressed can differ and the quality of the antibodies can differ. So sometimes you might have a really weak antibody, so you're going to get a weaker band even if you have more of the protein present than you would have with another sample. You also want to make sure that when you're analyzing the bands that you're not mistaking there for being more or something when you just loaded more of something. And so often what you also have in these lanes is you're testing for some sort of housekeeping gene or protein. This is going to be some that's like something that's constitutively and ubiquitously expressed. So it's expressed everywhere in basically all cell types, and it's used all the time. So the levels are fairly constant. This is going to allow you to normalize between different amounts that you loaded in different lanes. So you're typically trying to load really equal amounts between these different lanes. But sometimes your pipetting might be a little off or things like this. And so then you can kind of normalize things by dividing by this like lane normalization factor, which takes into account the strength of the band for one of those housekeeping proteins. So you might already have a lot of different things you're testing for, but one of them is probably you also going to want to test for the presence of a housekeeping protein. This is going to be important too when you're comparing your input to your pull down, because you might be using a diluted sample of your input in order not to overpower the pull down signal. And so in this case, you make sure that you're kind of taking into account that you have a diluted sample of your input. And this can then you'll see, so you should see a fainter band there in your input for that housekeeping protein. So those are just a little more technical details. It's really important too that you include a negative control. And this negative control is going to tell you about non-specific binding to the beads themselves or to the generic parts of the antibodies or things like this. So you don't want to see a band and think it's because your protein is binding to the bait, when really your protein was just binding to the beads. And so including a negative control can help with this. The negative control, there's going to be a variety of different options. So sometimes you might see naked beads used, so basically no antibodies attached to them. Sometimes you might see beads with antibodies targeting a different protein, or sometimes you'll see the same beads used, but in cells that don't have the bait. So for example, if you are having if you're using anti-HA, say, so it's targeting an HA tag protein, you can use this in cells with the normal version, the non-tagged version of the protein. So there shouldn't be an HA tag for it to bind to. And therefore, if you see things in your negative control lane, this is going to tell you that you're getting non-specific binding to that antibody. So you don't want to see bands in your negative control lane. If you do, then basically you want to make your wash conditions harsher, I'll get into. Or if you're worried about making your wash conditions harsher because then you're going to lose your actual bait, just at least keep an eye on those bands. Know that those bands, if you see them in your analysis, those don't represent true prey. They just represent these non-specific interactions with the bead and not specific interactions with your prey. So in terms of those wash conditions, you're probably going to have to optimize them. And so remember, the wash step is basically when you have your protein bound, then you're washing things off that aren't bound. And these are going to be like your wash buffers. Also, you're going to pre-incubate the beads with them. So before you actually add your mixture, you're going to get these beads acclimated to the wash buffer. So typically you do a few washes, and when you're talking about these washes, basically what you're doing if you're using the magnetic beads is you're just mixing those beads with the liquid, sticking those beads on one of those magnetic racks, sucking off the liquid, and then suspending it in more liquid, putting it back on the beads, sucking off the liquid. And so you do that a few times in order to get the beads ready. And then you'll do that a few times when you're doing the washes. And the wash buffer that you use is going to depend on what type of interactions you're hoping to detect. If you want to detect really weak interactions, so like friend of a friend, say, so a protein that's bound to a protein that's bound to your protein, or other sort of weaker interactions, you're going to need to use weaker wash conditions so you don't wash off those weakly bound things. But if you want to detect just the really, really strong interactions, so maybe just like the direct interactions and strong direct interactions, then you're going to want to use harsher conditions so you don't keep those loosely bound stuff. And so you don't have to risk there being this non-specific binding as much. And so the strength of your wash buffer is also going to depend on how strong the affinity of your bait to the beads is, as well as the affinity of non-specific binders to your beads are. So if you want, if you have like weak binding of the bait to your beads, you risk, if you increase the you harshen the wash conditions, you're risking that you're going to be losing your bait. If you have strong binding, you can afford to be a little harsher on the washing and not lose the bait, but you still might lose some of the prey. If you want, one thing that you can do as an alternative to, or not as an alternative, but in addition to doing washes, harsher washes, is what you can do is you can kind of like pre-clear the lysate. So you kind of, when we're talking about negative control, doing things like using beads that didn't have antibody, or beads with antibodies that targeted different proteins, and how we were doing that kind of like in parallel, so alongside our actual reaction. What we can do is we can add these beads before we add the beads of interest. And so in this way, we can kind of pre-clear things out. So we can get things to bind this decoy bait kind of, and then those non-specific binders, we can kind of remove that stuff, and then we add in our actual beads. So we basically mix our sample with those beads, the decoy beads. Then we take the liquid from that, and then we add that to the real beads. And this way we should have hopefully pre-cleared some of the things that bound non-specifically to those beads. And so this is a common way that people use in order to kind of reduce some of the signal from those non-specific interactions without having to get too harsh in their wash conditions. But say you do want to make your wash conditions harsher. Typically, this involves using a higher salt concentration and or higher concentrations of detergents or harsher detergents. So let's start with the salt. So a salt is just a neutral combo of a positively charged thing, a cation, and a negatively charged thing, an anion. So if you think of your typical table salt, that's NaCl, sodium is going to be your cation, and chloride is going to be your anion. And when you basically, when you stick a salt in water, often what they're going to do is they're going to dissociate. And so you have the positive and negative parts come apart. And now you have these ions floating around in solution. What's going to happen is the more salt that you have, the higher the what we call ionic strength, which is kind of like this charge fog. And so you have these charges are going to be surrounding the molecules, and opposite charges attract. So you're going to get the cations hanging out around negative parts of proteins, and the anions hanging out around positive parts. And what's going to happen is that these anions are going to, what's going to happen is that these ions are going to surround these molecules and make it harder for the molecules to find one another. And so they're going to be kind of binding to the salts, binding to these ions instead of binding to one another. So the higher the salt concentration, the higher the ionic strength, the stronger the interactions have to be in order for them not to be broken up. And so if you want to increase the harshness of your wash, remove some of those non-specific binders, some of those weak binders, you often increase the ionic strength, you increase the amount of salt in your solution. You can also increase the amount of detergent. So detergent basically is an artificial soap, and it's going to be able to kind of break up interactions. Often what we're doing is with these experiments, we're using some sort of non-ionic detergent. So something that's not charged is going to be, it's going to be gentler. It's going to not disrupt the protein interactions as much. Sometimes you might use harsher detergents if you want to use to do a harsher wash, or when you're doing the elution. So when you're actually getting things to come off of those antibodies, we often use SDS, which is an ionic detergent. And this is also going to be what we need in order to denature the proteins for our gel. And so this is going to be a harsh detergent. But typically for our washes, we're using some sort of milder detergent, something like Tween. So we optimize those conditions so that we don't get much in our negative controls. If we do get things in our negative controls, we know to keep an eye out for them. We make sure we have an input that we can compare things to, and then we compare the pull down to the input. It's important that we probe for the bait to tell us about the the recovery efficiency, as well as probe for different prey that we're interested in. But remember that it can't tell you the things that you're not looking for. So you have to know in advance what you want to probe for. And one of the last technical details I want to get into is that you might see bands corresponding to the antibody. So antibodies are made up of these two chains, this like heavy chain and this light chain, which are about 25 and 50 kilodaltons on an SDS page gel. And so if you run that gel, you might see a band corresponding at like a band at 25 and a band at 50. Depending on how clean your washes are and things like this, you might get a lot of bands still. But you'll see this 25 and 50. And even if you have a stronger washing, you still might see a band at 25 and 50. And it's likely going to be corresponding to antibody that you were using to actually capture that bait, especially if you're using antibody that's non-covalently bound to the bead. So it's not like permanently stuck on there, but it's bound through AG or something like this. Those bands might get in the way of your analysis. They might confuse you. And what can be especially confusing if you try to use your Western blot with a probe against the species that it came from, then you'll see those bands when you do the immuno blot. When you actually do that probing, you'll see bands there. So basically in all these antibodies, they have this unique part that's going to recognize the thing of interest and this kind of generic part. And that generic part is going to be specific for the species that was made it. So if those antibodies on the bead were made in, say, made in a rabbit, and then if you were used monoclonal antibodies to probe for something that were also made in a rabbit, and now you use a secondary antibody that's anti-rabbit in order to detect that primary antibody, well, now what you're going to do is you're going to be detecting those antibodies that were used to capture the bait, as well as detecting the actual protein, like the primary antibody that was bound to your bait in your probe step. So keep an eye out for that and try to avoid using the same species for those antibodies on the bead and the antibodies in your that you're using for your probing. So as you might surmise, based on all these caveats I'm telling you and about how you can get these like false signals and things like this, it's really important that you validate any of the interactions that you identify. There are various ways to do this. One of the most common ways and kind of most convincing ways is if you actually switch your bait and your prey. And so instead of using antibodies targeting the bait protein, well, now you use antibodies targeting the prey protein, which is now your new bait protein. So in this example, we would initially have antibodies that targeted this purplish gray protein. We would see if it pulls down the orange protein. Then we would take the orange protein, we would capture it, and we would see if it pulls down the gray purple protein. And so in this way, you might have to then go and you might have to label the orange one and stuff like this, so it can take more work, but it could be a really convincing way in order to show that these are actually interacting. Sometimes if it's a weaker interaction, you might not actually be able to see that it's going to pull down that other protein. It's going to be especially true if you're dealing with a non-direct interaction. So maybe this is bound to a protein that's bound to this protein. And only some of the time is that protein actually part of that complex. There are a variety of complicating factors. You can also have sort of indirect binding or binding that's facilitated through nucleic acids. So if you have like a DNA or RNA binding protein, the interaction between these two might not be direct. It might be that they're both bound to the same site on like an RNA or on DNA. To test for this, you can actually introduce a nuclease, so introduce like an RNase or a DNase in order to degrade that nucleic acid and see whether that interaction still holds. So you often see with experiments where people are dealing with nucleic acid binding proteins, they'll show a lane with like plus RNAs and minus RNAs or something in order to show you whether or not this interaction is direct, or at least it's protein-protein, or whether it's kind of mediated through this nucleic acid. Another caveat to keep in mind is that sometimes the interactions that you see might not be actually physiologically relevant. So they may not actually be things that are naturally happening. Typically in these experiments, you're dealing with some sort of cell lysate. So basically you break open the cells and then you bind things. And once you've broken up into cells, the conditions are going to be different from the conditions inside of cells. So inside of cells, these proteins are often kind of like partitioned off into different places in the cell. There's various things like this where proteins are kept together that are working together, and you have all sorts of different things that are kind of helping compartmentalize things inside of the cell. And then you break the cells open and you kind of make these all interact. And so proteins are going to be seeing proteins that they don't normally see, or they don't normally see in those concentrations and things like this. So this can work either way, where you're introducing false interactions, or you could be losing interactions because now you're lowering the concentration of the protein around that original thing. And so sometimes the interactions that you detect aren't actually interactions that you see inside of cells, or you're missing interactions that you see inside of cells, but you're not seeing here. So sometimes what you might want to do is also use a validation method where you're actually testing things inside of cells with some sort of proximity labeling, or some sort of immunofluorescence, or things like this, various different ways in order to actually verify that these interactions are real. And if you don't see those interactions, to see whether you actually do see them inside of cells though. Since you're using a lysate, you also have the option of kind of introducing a bait into that lysate. So instead of actually having to genetically engineer a bunch of different cell lines, or to introduce a bunch of plasmids into cell lines, you can kind of make and purify different versions of bait that you want to test, and then mix that test in with your lysate. So again, this is going to be less physiologically relevant than if you're actually trying to detect interactions that were happening naturally inside the cell, and you have to worry about whether you're adding too much of this protein. If you're kind of, the more of the protein that you have, the more kind of far away from the natural conditions you have, the more likely you are to get interactions that aren't naturally happening. But this, if you use these different, you can add this purified protein, see if it can bind, not necessarily whether it does normally bind, but whether it can bind. This can be important experiments if you're trying to do things like see whether a certain part of the protein is important for binding. And so if you're often using a cell lysate, but you can also be using purified proteins. There are a bunch of different variations to pull down experiments into co-IPs and things like this. And it all depends on what type of questions you're trying to answer. So are you trying to answer, we know that these interact, but which parts of the protein are important for interacting? Or we know that they interact, but does this interaction differ between different cell types? These are different types of questions that you would be asking in either cells, or you could be asking in just purified components, or just in the lysate. So lots of variations and lots of options. But to review, with any sort of these pull down assays, what you're doing is you're trying to capture a protein or some other molecule of interest, your bait, and then see what it's bound to, your prey. In order to do this, you're going to typically use beads that are going to be bound to something that can bind to your bait. And so in the case of a co-IP, this thing is going to be an antibody that binds to your bait. But there are other forms where you can use things like a glutathione tag, or streptavidin and biotin and things like this. You just need a way in order to pull down your protein of interest and isolate it from everything else except for the things that it's bound to. So typically what you're doing is you're going to be binding, you're going to kind of mix your sample with the beads. You stick these beads into, you either centrifuge them if you're not using magnetic beads, or if you're using these magnetic beads, you stick it against a rack. Your bait protein bound to the beads, and whatever the bait protein was bound to is going to get pulled to the side of the tube, or it's going to get pulled to the bottom of the tube if you're centrifuging. You're going to pipette out the liquid, and this liquid should be the bulk of whatever was in there that wasn't bound. But it's not going to be all of it, and so there's still going to be some stuff that's non-specifically bound. So you're going to be doing a bunch of several wash steps. Basically, you're going to be mixing your sample with some wash buffer, and then isolating your beads again, sucking out the liquid, doing that a few different times. And then what you can do is you can elute your sample depending on what you're hoping to do and what you're hoping to, what type of beads you're using, things like this. There are different methods. You might just heat it up and add your protein loading dye. You might use an acid or a base method and then neutralize it, various options. And so look to the manual and various technical specifications for your experiment. But in this way, you're able to then elute or kind of like unbind the stuff that was bound, and then you analyze it. And when you're doing that analysis, make sure that you have an input lane and that you have a negative control. And then you can probe for various prey that you think might be present. If you want an unbiased look at whatever was present, then you can use mass spectrometry. If you're looking for proteins, sometimes you might be looking for DNA or RNA that was bound, and so then you can do things like take that eluted stuff and then send it for sequencing. And so much more on various aspects that I went over in different posts, but hopefully this helps you understand the co-IP and other forms of pull-down assay.